So I will talk today about the importance of cutting-edge research to better understand and unravel the pandemic diseases that are present today. So, as you know, HIV came to us in the beginning of the 80s, and many clinicians would recognize the X-ray that is shown here as a picture of what we call a pneumocystis hyrovecii infection, where you see typical black holes as lesions in the lung, and these patients presented with a clinical picture accompanied by Kaposi sarcomata. So this was beginning 80s, and subsequently drugs have been developed, Cydovedin as the first one, and now we have a picture where we see patients that can be treated with one tablet once a day. So there's been an enormous revolution by in-depth research. Now, the discussion that I will bring today is how can we add to this and the experience and the research that we can do from multiple centers, and I coordinate that research from the University Ghent. The whole story for HIV starts probably already around the end of the First World War, where animals, mainly non-human primates, chimps and sooty-mongoose in Africa, in Central Africa, carried a virus which is very similar to HIV. It's the SIV virus, simian immunodeficiency virus. Now, this came to us over the decades of the 40s, the 50s, the 60s of the last century, and it came very clear in the beginning of the 80s that there was a new clinical picture that was not recognized by infectious disease specialists at that time. And just to illustrate the feeling that was present at that moment, because we tend to forget that, I show you a small part of the movie Philadelphia. So people were suffering, they were anxious, and there was also a kind of a mass history about transmission, the lack of research, there was anxiety in the community, and the situation was actually resembling a situation with many unknowns as we have it today. Now, then an extensive amount of research has been done, both by university labs worldwide, but also by the pharma company, and we should not forget that. There were a big driver, a big power of force behind the drug development programs. And then we come to a situation today where we have 38 million people that are seropositive worldwide, but still only like about 20 to 22 million people have access to therapy. So there is a lack to access to therapy, and that makes that still 1 million people die every year. So that's one of the major concerns for people on a global level that deal with HIV. Now, the question that we want to answer was a very particular question, and I come to that in 1 minute. So here you see when a healthy individual, which has a lot of immune cells, when this person subsequently acquires an HIV infection, HIV is like rapidly spreading and multiplying, and that is depicted by the green green figures in the second person, and the amount of immune cells are decreasing fast as they are destroyed by the HIV. Now, when this person is treated with tablets, the virus is decreasing, and the immune cells can restore. So that's the basic of HIV therapy. You decrease to an enormous amount the viral load, the virus cannot replicate anymore, and the immune cells can restore. Now, when therapy is halted, the virus comes back, it re-emerges, and the immune cells are again destroyed. So, and one key question in HIV is, where is this virus actually coming from? Because we can decrease it to what we call undetectable levels, so it's basically not detectable, but when you stop treatment, the virus always comes back. And this was like looking at us trying to identify the needle in the haystack, where you want to see where is this virus coming from. So you see it here depicted. There is definitely virus, but we don't know where it's coming from. So to answer that pragmatic question, we initiated a clinical study where we first did a theoretical examination, thought, yeah, where do we have immune cells? Because it definitely comes from immune cells. So there are different anatomical compartments that harbor these immune cells. And as depicted here, you have in this pictogram, you have immune cells, for example, in the lung or in the gut. And these immune cells would probably have unique viruses because the virus has a high mutation rate. If the virus is different, probably in the lungs, than in the gut, than in the lymph nodes, than in the blood. So it has a unique sequence, which for simplicity we could call a unique barcode. And these viruses can hence be present in all tissues that harbor immune cells. Now, if you want to do a clinical study, you want to examine all these tissues, this is quite a heavy procedure. Why? You first obviously need to set up the clinical trial, you need ethical commission, you need patients to be on board. Patients in our setting, we designed this study in 2015, have co-developed the protocol to design a clinical trial where we could do a bronchoscopy, a lymph node excision, a gut biopsies, a lumbar puncture to analyze all these reservoirs. So they co-developed the procedure. We went to the ethical committee. They supported us. It was a very narrow interaction with patients. We did the procedures. We did a lymph node excision. That went subsequently to the lab. And there you have the whole path, because there it really starts. You have to separate the cells, to phenotype them, and then to come finally to a picture where you can analyze in the different tissues the viral reservoirs and the viral barcodes. When you subsequently stop treatment, this virus also has a unique barcode, and then you try to map the barcodes of the virus that comes back to the barcodes of the different tissues. That was the whole idea. We did that with 12 patients. We mainly analyzed 4 anatomical compartments, the blood, lymph node, gut, and the central nervous system by doing lumbar punctures. We, for many of these compartments, we looked at different cell types, and we then had, in this, per patient, we had more or less 360 barcodes that we analyzed, 360 viral sequences that we analyzed for 12 patients, so, making around 4,500 barcodes that we had in total over these 12 patients. And then we basically came to the conclusion that virus could be, the viral rebound, it's described there, is predominantly fueled by genetically identical viral expansions from diverse reservoirs. What does that basically mean? That the more cells that harbor a virus proliferate, the more they are probably linked to a virus that can come back, and this can be from any reservoir. Virus is randomly coming back. It's not from the gut, from the lymph node, or from the blood. It's in every patient different, and it randomly comes back, and it also is not from one particular cell type. It can be from different T-cell subpopulations in the patient. So it makes the picture more complex, but at least we also know that looking at one reservoir will not solve the problem. We have to attack it broadly, and that's where we're working on now. We are setting up a platform with collaboration in Flanders with other people to manipulate T cells to try to develop what they call CAR T cells, which are T cells that really recognize cells that are infected with HIV to go to next steps, and therefore you need a GMP production unit and so on. And this is what things that we are currently setting up at the University Ghent. Now, while all of this was ongoing, we had in March a huge COVID outbreak, a pandemic infection that was announced by WHO. So, and then we were thinking, yeah, we have to stop, reduce our HIV research. Can we help to support with the platforms that we have installed for HIV? Can we add knowledge to these key questions that are there? And what were key questions? Well, the diagnosis is classically made of a COVID by a nasopharyngeal swab, which is then PCR analyzed and can make a diagnosis. But some patients have negative swabs, but still have infiltrates, as you see here on the CT scan in the lung, and or like this picture also is showing, and they quite sick, so they have a clinical picture, but you cannot make the diagnosis, and that makes it very hard in a hospital. You have to decide, is this person going to go to a COVID negative ward or to a COVID positive ward? That's a crucial question you have to make every day. So we started to initiate what we were doing for HIV, to ask on the pulmonologists if they could do bronchoscopies under full protection. And that's what we, at the end, in a bronchoscopy, you do a lavage, and there you really go where the infection is, to try to amplify virus from there. And we have worked that out in a diagnostic platform, but what the bottom line is, in the 27 bronchoscopies that we did, 7 of them were positive. So 1 in 3 yielded still a diagnosis of a COVID infection. Now, these are some of these patients. So we had COVID positive and COVID negative patients. The COVID negatives had a clinical picture resembling COVID, and they served as controls for the COVID positives. And then we set it up a whole pipeline, well, because one of the challenging things was this came to us like a bomb. You know, you had to act. You knew patients were coming in the first wave. This would take 6 to 8 weeks, and then the first wave was gone. If you want to do sampling, you had to do it now and not in 1 month. So we had to act very fast. We wrote an ethical committee overnight, sent it to the ethical committee commission. They had procedures in place to accept that very fast in all university centers in Belgium. And in 48 hours, we could collect blood, PBMCs, plasma and bronchoalveolar lavage, and then allocate that to answer very particular questions, but also to look broad. One of the protocols that we performed was SiteSec, where you really in depth analyze every cell in the lung present at this certain disease stage and look to the RNA expression pattern, the protein pattern, and then you can try to learn. Here we learn, for example, by this protocol, that we can have certain genes in populations from B cells that, certain genes that are related to certain activities. For example, here you see a pattern, and I'm not going to go in detail, but what it shows is that B cells start to proliferate very strongly in the PBMCs, and you can then link that to what you see in the bronchoalveolar lavage in the lung and in the blood. You can try to map that. You can do pathway analysis and really try to better understand the pathophysiology of the disease. And this we only could do because we had a platform from HIV in place. So when I'm going to conclude here, cutting-edge research to unravel pandemic disease. What is important? Well, for us, as a clinical research, in-depth sampling really paved the way. This was a research question that has not been answered, but by combining all these tools, doing bronchoscopy, lymph node excision, having all disciplines on board, we could finally answer a key question in HIV research: where is this virus coming from? And it appears to be random and from multiple organs. Now, the patient involvement is crucial, and that is really also for the COVID research. You know, you have to have patients on board because you need in-depth samples. You need to explain it, and this takes time. If you have patients where you have to explain an in-depth protocol, you don't do that in 5 minutes, and you have to do that on your own. You cannot ask a junior doctor to go and explain a complex protocol. These are things that are very time-consuming. Quality is for me much more important than quantity, so you have to have high quality samples, not 1,000, but you have to have high quality samples if you want to learn something. Now, and COVID research is currently moving very rapidly, and I think we all a bit skeptical that sometimes controls are skipped. So adequate controls should be taken into account, and that is true for every step. It can be basic research, translational research, vaccine development. The controls are very important because you have to always balance what you see versus controls that are COVID negative. And one of the things that we learned from HIV along the way is that social science have supported HIV research dramatically. By better understanding how people have to deal with it, they are more involved in research and they help supporting your research. And that commitment should come along also for COVID-19. We have to explain that very well to the population, to the people, that this is a very important step to make, and it will only be research that been developed that give us new tools, new drugs, new vaccines that will help solving that problem. It's not going to come from somewhere else. It's the research that will have to do with the biomedical science, and that's why I'm very proud on the research that we could do, because we could implement HIV platforms to COVID research, and now these samples are used in worldwide analysis with collaborations with the US and others to be further looked at and searched for new therapeutic tools. Thank you for your attention.